Hello dear students, welcome to another tutorial from Brilliant Bios. This is my cellular energetics lecture series. In this video we will learn about the structure and function of complex 1 in electron transport chain in more details. In my previous video I have already discussed about the basic concepts of electron transport chain and protein motive force. So now in this class we will learn about in more details. As I have already mentioned in my previous classes that TCA cycle and the electron transport chain actually occurs in the mitochondria. So here inner mitochondrial membrane is important because all the complexes that take part in the electron transport chain are embedded in the inner mitochondrial membrane. So let me draw a inner mitochondrial membrane. Let's suppose this is the inner mitochondrial membrane. So this is inner mitochondrial membrane, and let's suppose this side is the matrix side and this is the intermembrane space. Okay, the shape of the complex one is like L, alphabet L. So let me draw the structure of the complex one. By the help of electron microscopy and X-ray crystallography, scientists have found that in the bacteria there are 14 subunits. So in the bacteria there are 14 subunits present in the complex 1, and they found that these 14 subunits are essential for the functioning of this complex 1. So this is the minimum number of subunits that is required. But when they discover the structure of complex 1 in the eukaryotes, they found that there are also 14 subunits that is structurally similar, actually conserved, and those actually resembles the bacterial counterpart. But along these 14 subunits there are 26 to 32 accessory subunits are also present. So in case of eukaryotes, the complex 1 contain more than 40 subunits. So these are called accessory subunits. You have to understand this. Now the name of this complex 1 is called NADH. So I am writing here NADH CoQ means ubiquinone reductase. So what happens here? I have mentioned in my previous classes that the NADH is the high energy electron carrier that carry one pair of electrons. Now this complex, actually the site where this NADH actually go through oxidations. So what happens here? NADH actually release its energy and become NAD plus and release two high energy electron in this complex. Okay, I am denoting electron, high energy electron, as red color. So scientists have found that in this domain, this membrane embedded domain, there are 4 subdomains are present. Among them, 3 subdomains have the property like cation antiporter. So there are 4 subdomains present. Among them 3 subdomains of this plasma membrane embedded portions, subdomains, they act like a cation antiporters, and this is very important points to remember for the complex 1, because complex 1 indirectly helps in the generation of proton motive force, that means transportation of protons or pumping of protons. So there are 3 subunits that particularly helps in the proton pumping. Now the important fact about this complex 1 is the presence of some prosthetic groups that can carry high energy electrons that are released by the NADH. The first prosthetic groups that actually carry the electron are called FMN, flavin mononucleotide. So this compound can carry electron, but here this FMN and all the subsequent prosthetic groups actually carry one electron at a time. So you have to understand this fact. NADH carry 2 electrons, high energy electrons. They release the electrons in this complex 1, and the first acceptor of this electron is the FMN, but they can carry 1 electron at a time. In this complex 1, there are 7 ferrous sulfur clusters are present. These are also prosthetic groups that can carry electrons. So 1, 2, 3, 4, 5, 6, 7. So there are 7 FES, that means ferrous sulfur cluster, and all these 7 ferrous sulfur clusters and FMN are togetherly called redox center, In my previous class, basic concept of electron transportation, I have already mentioned a term called redox potential. You have to understand this thing very clearly. Another time, I have mentioned a term called redox potential, that is similar to the electron affinity. Okay, so what I have mentioned that during this transportation, because this is called electron transport chain, during these transportations, electrons actually jump from one prosthetic group to another prosthetic group, and this direction of the transportation is actually unidirectional. It actually goes in one direction, it cannot reverse. And the reason behind this is that all the prosthetic groups that actually progress, they all have higher redox potential or electron affinity from their counterpart. That means this FMN should have higher redox potential than NADH. This ferrous sulfur cluster should have higher redox potential than this FMN, and this is the series. So the redox potential should be low to high in all these prosthetic groups, so that electron flows downhill movement, okay, and in unidirectional. You have to understand this. So this redox centers has one property you have to remember, that is all the prosthetic groups that are present have the low to high redox potentials. And the second property, this is the first property, and second property is the distance. The distance between two prosthetic groups should be ideal for the jumping of electron from one molecule to another. So distance should be ideal, and the distance is really ideal for electron jumping. So one electron jump from one prosthetic group to another prosthetic group, but it cannot jump from one to another because the distance is actually prohibited. So it cannot jump from one to the next before escaping the second. So it is not actually permeable, so it is prohibited. So electron jump from one center to another center, and by this way it reach to the ultimate electron acceptor in this complex one is called CoQ. The name here NADH CoQ reductase means here NADH actually gets oxidized and CoQ gets reduced. So let's suppose this is a CoQ that ultimately accepts, so this is the CoQ that ultimately accepts the high energy electrons. Okay, so electrons are moving from this one at a time in these directions, and this CoQ can accept 2 electrons and can become CoQ to CoQH2. CoQH2. I am writing in this membrane CoQH2. Why? Because this CoQ means ubiquinone. CoQ means ubiquinone is actually a lipid soluble molecules that can actually go through the plasma membrane. So this molecule can actually pass through the plasma membrane, and ultimately this CoQH2, this ubiquinone, reach to the complex 3. So this is the whole story. So these 2 points you have to remember. Now the important point is how this structure the complex 1, helps in the transportation of protons. Helps in the transportation of protons because it actually pumps protons. 4 protons actually transported. 1, 2, 3 and 4. So 4 protons actually transported out for each pair of electrons. You remember, each pair of electrons. And these all prosthetic groups actually transport electrons one at a time. Now it has been found that during these transportations, electron undergoes a drop in potentials. You have to remember this. During these transportations, electron undergoes a drop in potential of about 360 millivolt, and the delta G0 bar, that means free Gibbs energy, is approximately minus 16.6 kilocalorie per mole for 2 electrons. So you can see here, by moving or transporting this electron, it releases energy, and much of the energy actually used for the transportation of protons by the help of changing the conformation of the subunits. So during these transportations, electrons actually when the electrons undergoes these transportations, potentials difference occur approximately 360 millivolt, and if you calculate the delta G0 bar, then you will find that approximately 16.6 kilocalorie per mole for 2 electrons. So that much of energy is actually used by this complex to change the structure of the molecules. But I want to mention that the structure of complex 1 is still now not completely known. So the mechanism, how the structure, the conformational change occurs and protons transportation occurs, is not completely yet known. So we know that there are 4 protons actually transported out. Now I have mentioned that this coenzyme Q can be co-QH2, that means it can actually accept 2 hydrogen atoms, and the 2 hydrogen atoms actually come from this matrix site. 2 hydrogen atoms actually attach with this CoQ to make CoQH2. In my next class I will talk about the complex 2. So hope this video will help you. Thank you for watching, and always keep learning.